Welcome back to Drugs and the Brain. This is the first lecture that we will actually go into depth on our subject matter, and we are going to discuss the concept, what is a drug, by giving you examples of 4 drug molecules today. First of all, though, we want to talk about the origin of the word drug. It arose in Middle English in about the 14th century, and we've gone to a dictionary to get a good series of definitions for drugs. First, in general, it's a substance used as a medication or in the preparation of medication. About a century ago in the U.S., the Food, Drug, and Cosmetic Act systematized the definition of a drug, at least for regulation by the FDA, by the Food and Drug Administration. It's a substance recognized in an official pharmacopoeia or formulary, and you can see 2 pharmacopoeias, one for animals and one for people, at the FDA, fda.gov. According to that same act, it's a substance intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease. And also, it is a substance other than food intended to affect the structure or function of the body. So, food is not a drug, an irrelevant definition. And finally, according to the dictionary, a drug is something, and often an illegal substance, that causes addiction, habituation, or a marked change in consciousness. And this is the definition used by many people, and it is also the definition used by the U.S. National Institute on Drug Abuse, part of the NIH, which supports a lot of research, including some of my own research. In terms of trivial names for drugs and structural formulas, we're going to give as an example nicotine, and throughout this course we're going to use nicotine as an example. It figures prominently in my own research. We will also discuss lidocaine today, morphine, and also botulinum toxin. These are very different molecules, as we'll see. The first 3 of them are best described in an NIH database called PubChem, and here are the accession numbers for each of the records. So if you type PubChem 3676 into your search engine, you'll get the record for lidocaine, etc. Botulinum toxin is best described in another database, the so-called Protein Data Bank, whose files are curated by an international organization that looks at X-ray crystallographic coordinates and at nuclear magnetic resonance structures. So these are our 4 examples, and they exemplify the concepts that we are going to discuss in Drugs and the Brain. Drugs activate and block ion channels. Nicotine activates, lidocaine blocks ion channels. Drugs act on G-protein pathways, for instance, morphine. Eventually, if an organism is exposed to drugs, both nicotine and morphine activate genes, an important concept in chronic exposure to drugs. Botulinum toxin is an example of a protein drug, and this class may become more useful for neuroscience diseases eventually. Botulinum toxin is really the only example at the moment. In this session, we won't treat drugs that act on neurotransmitter transporters, but they are very important, and we will treat them later on in the course. In terms of atomic scale structures, it is perhaps useful to go to PubChem, to the record for nicotine, for instance. What do we see here? We see that the nitrogens are in blue. This is more or less standard. And to get the best possible dimensionally accurate structure, we go to the 3D conformer section, and there are actually about 8 of these conformers in PubChem. They have rotations around this single bond. They have slightly different puckering and torsions and angles on all of the bonds, and we can get a good feeling for the ways that the nicotine molecule can change its shape. We can do the same for lidocaine. Here for lidocaine, we add the fact that the oxygen atom is usually portrayed in red. To get a nice structure of botulinum toxin, we would go to the protein data bank, and we will do so in subsequent lectures, but not today. One of the ways that we can download from PubChem is a program called PC3D, and it is a very beautiful way of showing a drug in greater detail. It allows us to point out that each moiety, each group of atoms in a drug molecule, has importance for the function of that drug. Here in this example, lidocaine, lidocaine has a charged amine group, which may bind to oppositely charged groups, negatively charged groups, or to pi electrons on the receptor protein. It has an amide moiety, which typically is hydrolyzed by endogenous esterases to terminate the action of the drug. And this termination occurs more slowly for an amide than for an ester, and gives us roughly the right amount of time for the local anesthetic to act, an hour or so. Most drugs like lidocaine have aromatic groups which can bind to nonpolar groups on the receptor protein. In addition to these moieties, medicinal chemists tune up the function of a molecule by adding substituents such as alkyl groups. Here are two ethyl groups on the amine. This affects the charge density, which in turn affects membrane permeation and binding to the receptor. And likewise, it's possible to tune up the properties of the aromatic group, here with methyl groups. So this is a wonderful set of tools that the pharmacologist and the medicinal chemist can use to make the best possible drugs. In terms of atomic formulas and molecular weights, here's a list. The unit of molecular weight, also called molecular mass, is the Dalton, which is the same thing as the number of grams per mole, and to a very high degree of accuracy, also the same thing as the number of atomic mass units. So nicotine has a molecular weight of 162 Daltons, lidocaine a bit more, morphine a bit more, botulinum toxin very much more than that. These are typical molecular weights for the drugs that we will discuss, up to about 500. Now, let's talk about ionization state in counterions for a moment. Probably the best way to talk about this is to go back to the PubChem entry database and type in nicotine, and we will find not one but several records for nicotine. Here is the one we were looking at previously today. Here is one in which the molecule also contains two tartrate ions, and here is one in which we have a green and a gray, and if we expand this, we find that the gray is a proton, and typically in this chemical, the proton is non-covalently interacting with one of the nitrogens and gives the molecule a permanent positive charge, and as a result, in the salt, there's also a chloride ion. Now, no natural source of nicotine and no nicotine-containing product, such as a cigarette or a gum or an electronic cigarette, has this salt version of nicotine. They all have the version that we discussed at first. But in my laboratory and in other research laboratories, we often do buy this salt from chemical manufacturers because it is nice and soluble, and it allows us to define concentrations very accurately for experiments on isolated cells and organs. On the other hand, when one buys lidocaine or morphine from the manufacturer, these are salts, soluble, sometimes injectable. So typically lidocaine is found as the hydrochloride. Morphine is found either with a proton attached with sulfate as the counterion or with chloride as the counterion. So the molecular weights of the total complex here would be higher in each of these cases. This is a complication that people know about and use. Botulinum toxin presumably has in it protonated and unprotonated groups. It may have some chloride or sulfate ions. It may have waters of hydration in it. Counterions and waters of hydration are rather poorly defined, as far as I know, for botulinum toxin. We're not going to use trademarks in this course. The reason is that they vary by country, by the intended use, and by the counterion. Here are some examples of trademarks for nicotine in cigarettes, or in electronic cigarettes, or in gum, or in patches. There are many different trademarks for lidocaine depending on the use, whether it's on the skin or injected, which country it's being sold in, and which pharmaceutical company is selling it. Likewise for morphine, whether it's being used as an analgesic or a sedative or as an adjunct to anesthesia. And botulinum toxin, when it's used as a cosmetic, mostly has this trademark, but other manufacturers and other countries provide it with other trademarks. So we won't use trademarks at all. In terms of types of compound, we often refer to nicotine and morphine by the somewhat old-fashioned terms alkaloids. Alkaloids are a group of nitrogen-containing compounds, typically from plants or fungi, usually natural sources. And mostly alkaloids are drugs. Lidocaine, even though it has a nitrogen in it, is more properly called a local anesthetic, and it's a synthetic organic compound. It does not exist in nature. It's a derivative, it's an analog of procaine, which in turn is an analog of cocaine. A rather interesting story from the early days of medicinal chemistry. And finally, botulinum toxin is a protein, which is a chain of amino acid residues joined by peptide bonds. So we see there are many different ways of describing drugs. We've described a few of them in this session. Next session, we'll talk about how drugs enter the body.